Hi guys, so today we're going to be doing an overview of cyclin is degraded by the ubiquitin pathway, and this is by Glotzer et al. And was published in 1991. Okay, so here's our overview. Proteolysis is a critical part of mitosis. Proteolysis is the hydrolysis of the peptide bonds that hold proteins together, and this results in the breakdown of proteins into their key components, peptides and amino acids. Mitosis is induced by activation of M-phase promoting factor, also known as MPF, which is a protein kinase, and it has specific subunits that have extensive homology to certain known protein kinases and cyclin. The metaphase to anaphase transition is induced by cyclin degradation. This leads to the inactivation of MPF, and many things must occur in order for another round of mitosis to occur. So these things include a decrease in cyclin degradation, cyclin accumulation, MPF activation, and only after all of these things occur can the next round of mitosis start. Regulation of cyclin degradation is now well understood. It is the crucial event in the exit of mitosis and entrance into interphase, and cyclin mutants that cannot be degraded arrest the cells in mitosis. It is clear, however, that cyclin proteolysis is triggered by the presence of active MPF, and once it is initiated, it's very rapid and highly specific. A prominent degradation pathway is mediated by the conjugation of ubiquitin, and this topic is relevant to all of us because we recently learned in class about polyubiquitination and the role it plays in many pathways in our cells. Okay, so on to our methods. There was a bunch used in this research paper, and so we have expression and translation vectors, SDS page, audio radiography, mitotic extracts, radio labeling, kinetic analysis, and pulse chase analysis. And the cells that were used were xenopus cells. Okay, so on to our results and figures. Here we have figure 1, and a protein synthesis requirement of early xenopus cell cycles, is fulfilled by translation of sea urchin cyclin B mRNA. Here in figure 1, a truncated cyclin was used. Cyclin 90 lacks the first 90 amino acids. It is not degraded at mitosis, but it is still able to activate MPF. This was used to induce the stable mitotic state to dissect the cyclin destruction pathway and characterize the cyclin domain. Figure 1 shows the effect of adding cyclin 90 to interphase extracts, and it also shows histone 1 kinase activity, which is a measure of MPF activity. And as you can see here, this peaked at about 30 minutes after the addition of cyclin 90 and remained high throughout the rest of the time. Cyclin was first detected up here between 40 and 50 minutes, which is right here, after the cyclin 90 addition. Cyclin that was added at later times was also degraded, showing that cyclin proteolysis activity is maintained at high levels for at least an hour. Without added cyclin, the extracts remained in interphase where H1 kinase activity is low and cyclin is stable. Okay, so here we have figure 2. Previously, it was shown that amino acids 13 through 90 of sea urchin cyclin B are essential for cell cycle regulated destruction. Here in figure 2, many hybrid proteins were constructed and expressed in E. coli and then purified through affinity chromatography and radioiodinated in order to be evaluated. As you can see here in C and D, amino acids 13 to 91, protein A, were efficiently degraded in mitotic extracts, but were stable in the interphase extracts. This means that the residues 13 through 91 of cyclin contain sequences that are sufficient for cell cycle regulated proteolysis. A, B, and G show the results of a test to see if the structural elements have a functional significance. In these, the residues 66 through 98 were deleted from cyclin 13, which resulted in a degradation in mitotic extracts and stableness in interphase extracts. A deletion of 54 through 98 produced stability in both mitotic and interphase extracts. This figure also shows the results of fusion of the 13 to 66 residue to protein A, and it was sufficient for proteolysis, but 13 through 53 was insufficient. Therefore, the 54 through 66 residue contained a determinant that is required for cyclin degradation. And then 2G also summarizes the behavior of the mutant constructs. Okay, so for figure 3, here the relationship between cyclin proteolysis and exit from mitosis is studied by testing the ability of cyclin 13 R42C to drive extracts in and out of mitosis. Accumulation of cyclin 13 R42C and control cyclin 13 activated histone 1 kinase and caused nuclear envelope breakdown and condensation of chromosomes. In the cyclin 13 containing extract, cyclin degradation was induced 40 minutes after the beginning of the reaction, and H1 kinase activity decreased shortly after. So you can see around here, this is the 40 minutes after when it began to degrade, and you can see that the kinase activity down here also decreased. Okay, so for figure 5, here we're looking at an autoradiograph of the affinity purified 13 through 91 protein A and conjugates. Lanes 3 and 4 show a comparison of the ladder of labeled bands that were precipitated only when the 13 through 91 protein A was present. These experiments show that the conjugation of ubiquitin to cyclin derivatives is regulated during the cell cycle. This also shows us that multi-ubiquitination of cyclin, is required for destruction. Okay, so for figure 6, this was an attempt to demonstrate that at a steady state, the flux of cyclin through the ubiquitin-containing intermediates is equal to the overall rate of cyclin degradation. To model this, a pulse-trace experiment was carried out. As you can see, this figure, the radioactivity of ubiquitin conjugates decayed to less than half the original amount within 120 seconds. And 6B, right here, compares the values of the concentration of unconcentrated cyclin and cyclin ubiquitin conjugates from earlier experiments. The close agreement that is displayed between these lines suggests that all of the cyclin that is degraded passes through a ubiquitinated intermediate. Okay, and now for our results and discussion. Here are some of the most important results from the study. The N-terminus of cyclin is necessary and sufficient for M-phase-specific degradation. Cyclin degradation at mitosis is accompanied by the formation of cyclin ubiquitin conjugates. A mutation within cyclin that inhibits degradation also inhibits ubiquitin conjugation, and MPF regulates conjugation of ubiquitin to cyclin, leading to rapid destruction of cyclin by a constitutively active system. The results of this experiment helped to create a pathway for further research on cyclin degradation and associated pathways. Some of the research that can be taken further is the difference between A and B cyclins. More information can definitely be found out about the destruction box. The structural requirements for degradation can be looked further into, and a large number of research regarding regulation of the ubiquitin conjugating system by MPF. Okay, guys, thank you so much for listening, and I hope that helps you guys understand the paper a little better. Thank you.